Hello, everyone. We're going to get started. I'm looking forward to our amazing discussion to have. We have a great panel of speakers as well as moderators for this session. This is the Oncobiome session, so if you're in the wrong session, now is a good time to leave. We won't judge. My name is Laura Bukavina. I am a recent Fox Chase transition graduate of fellowship, now transitioning to Case Western as an assistant professor in urologic oncology. And I am incredibly interested in microbiome, particular modulation, and how in bladder cancer patients we can really modulate it on multiple levels. So this Oncobiome session is for you. This session is planned to have a whole spectrum of results, both nutritional, federal regulations, probiotics, as well as emergent basic science, translational, and patient outcomes data. So I am going to introduce our first speaker. That's Dr. Leigh Greathouse, right in front of me here. She does amazing work in nutritional sciences and microbiome from Baylor University, and I'm going to have her take over and start the presentation.
Thank you so much for inviting me to speak today. So today's talk, I'm really going to try to focus on where I think our profession needs to be going in the future. So I'm going to kind of lay it out. Sure. I'll talk until that happens. Okay. Okay, perfect. There's one up here. Okay, I'll just wait until my slides come up and I'll keep talking. So my talk today is really going to be kind of a pilot of what I would envision kind of my TED Talk is, because I'm very passionate about trying to move our field forward into thinking about precision nutrition therapy or precision nutrition medicine. It's kind of set in the back end or the back mind of our thoughts when we think about treating patients, mainly because there's just not significant data out there right now that's clinically relevant that you would want to implement into practice. But the way I'm going to lay out the presentation today, what I hope to start to put into your mind is some of the evidence that we have that might support putting precision nutrition therapy as more of a first-line component of therapy. And some of the things that we need to think about going forward when we set up these research projects, when we do nutrition intervention, because it's languished because we just haven't had enough well-designed, large nutrition intervention studies with a lot of evidence-based. So my slides are up, so I'll go ahead and get started. All right, so this is my disclosure for today. And like I was saying, my proposal, thank you. Thank you. Appreciate it. So what I want to propose today is that the combination of precision nutrition therapy plus precision microbiome therapy would lead to improved treatment outcomes for cancer patients. And what I'm going to do is set up the evidence for that and then lay out what research we need to move forward into this area. But first, I'm going to tell you my story, especially for the cancer patients that are online, because it's really important to help you understand how I got to this point. So during my master's degree, I was doing exercise and sports nutrition. I was diagnosed about halfway through with a really rare form of cancer, uterine leiomyosarcoma. It was a stage 4. And thankfully, at the next end of the year of 99, I had a clean CT scan. Yay. And what I realized during this cancer treatment process is that when I changed my diet radically to eat a very high plant-based diet, eliminated a lot of processed foods, my energy levels increased dramatically. And this kind of was in line with I was already studying in nutrition. And so this made me really passionate to find out more about this interaction with cancer prevention and cancer treatment outcomes in nutrition. So I finished my degree in exercise and sports nutrition and went on to get my PhD in molecular carcinogenesis to start my research endeavor. During this time, I got into the NCI Cancer Prevention Fellowship, where I fell in love with the idea of the microbiome and its modulation between diet and cancer treatment response. So I hooked up with Julie Segre and Curtis Harris, and we started a cancer microbiome research project. which culminated in some really interesting findings in lung cancer. Now, when I started my own lab, I wanted to combine my love and passion of nutrition along with the gut microbiome to identify tools and factors and mechanisms that might improve cancer treatment response for cancer patients so they didn't have to suffer a lot of the same side effects that I had decades to suffer with. So that is the goal of my research lab, is to identify tools and mechanisms to help improve cancer treatment response. So first, I'm going to start by laying out what is the evidence that the diet and the gut microbiome can actually change health outcomes in general. So one of the studies that I love to talk about is this one from Jeffrey Gordon's lab back in 2013. And this is what kind of really invigorated my research passion for the microbiome. So they looked at twins that were discordant for obesity, and they did a fecal microbiome transplant into germ-free animals, feeding them critically the same exact type of diet. And so what happened was that these germ-free mice took on the phenotype of their human FMT donor. And so what happened with the mouse that got the microbes from the obese twin, or the twin with obesity, had increased adiposity. And we now know that's because it turned on genes that were associated with increasing of fat deposition. But interestingly enough, so we all know that mice and other animals are coprophagic. When they put them in a cage together, it was critically important what type of diet they were on that allowed these microbes to colonize the gut. So if they were on a low-fat, high-fiber diet, the animal that had the microbiome from the lean was able to colonize the microbiome from the obese donor. So diet was a critical component for colonization of these microbes. So I explained just recently that the diet could be a component of microbiome mediation in these health outcomes. But since then, several reviews and other studies have shown or demonstrated that the diet is a very strong component, if not one of the strongest components, besides of region of where you grew up, in dictating what kind of microbes colonize there and how they metabolize the different kind of products that we take into our body and we're exposed to. So we know that obesity and physical activity are one other two strong phenotypes or activities that change the microbiome. But on the opposite side, our diet, being either prudent, meaning high dietary fiber, can induce changes in microbes producing short-chain fatty acids, which control our immune system and inflammation, and a more westernized diet that lacks dietary fiber, high in dietary fats, processed meats, induces or encourages the growth of microbes that produce metabolites that can induce DNA damage through production of secondary bile acids or hydrogen sulfide or trimethylamine oxide. So these types of bacteria that metabolize our dietary products can control immune response in our entire body. So one of my favorite examples of this is from a study by O'Keefe in 2015, and this is demonstrating how quickly diet can change these markers of cancer, at least for colon cancer prediction and prevention. So in 2 weeks, they did a dietary exchange between Africans, rural Africans, and African Americans. And they did this because the incidence of colorectal cancer in rural Africans is only about 5%, 5 out of 100,000 people, whereas in African Americans, it's extremely high, over 60%. So if you notice the difference between the diets in the African Americans and the rural Africans, you know, you can see that the fat is different, the carbs are different, but what is dramatically different is the fiber intake. So, and among African Americans, it's only 14 grams in their entire diet. It should be more like 28. In the rural Africans, it was 66 grams of fiber. Now, I don't think most of us can actually get that much fiber in our diet. But their main type of dietary intake was a type of root vegetable. And when they did colonoscopies at the end, even though the rural Africans had quite a bit of parasites in their colon, they still have reduced risk of colon cancer significantly. But in just 2 weeks after changing the diet, the African Americans had a decrease in biomarkers of cancer risk, looking at the Ki-67 staining. They also had increase in butyrate, the short-chain fatty acid, and decreased secondary bile acids. And this is just after 2 weeks of a dietary exchange. So it can change the microbiome dramatically if you do change your diet dramatically. So overall, diet can impact risk factors, at least with colorectal cancer. But my question is, can you use diet to actually impact cancer treatment outcomes in the short term? So, can diet affect the microbiome enough to impact cancer treatment outcomes and survival overall? So that is what I'm going to start talking about next. So the first paper I want to introduce is one from Jennifer Wargo's group at MD Anderson. Maybe some of you are familiar with this. It kind of launched our thought in this field that the microbiome might be related to cancer treatment outcomes. So in this study, they looked at the microbiome, the fecal microbiome, and the oral microbiome prior to the start of immunotherapy, and then again at the end of this therapy session. And so on the top is the oral microbiome in the bar chart, and the bottom is the fecal microbiome. Actually, that's reversed. Oral is on the bottom. So in this study, they see initially that the diversity of the microbiome seems to be associated with response. And I say seems to because I want to put a pin in that for now, and we'll talk about that in just a second. So they initially thought that the diversity or the composition of the microbiome in your gut was related to response to immunotherapy. And what they looked at specifically was a certain bacteria called Rhamnococciaceae. It's a family of Rhamnococcus. And what they were seeing is that this specific bacteria seemed to be more related, or the abundance of it seemed to be more related to responders. In the other two charts, you're seeing the survival associated with high diversity in the blue, and then moderate or low diversity in the red and the gray. So they initially thought it was diversity, but they had this initial clue that Rhamnococcus might be a component of it. So fast forward to 2021, I was really finally excited to see this work because I saw it as an abstract, I think, in 2019 at ASCO. And I knew this was coming out, and I was super excited when I saw it. I think it was around Christmas I saw it, and I was super excited to read it. So what they were looking at, and it was a really great study, very, very well designed, large numbers. They had two different cohorts, one of melanoma patients and another set where they were specifically looking at dietary and other lifestyle factors that were could predict response to immunotherapy. And so they looked at specifically dietary habits and probiotic use in combination with immune checkpoint blockade. Importantly, they saved the stool samples from these patients and inoculated germ-free mice, so they did fecal microbiome transplants. And from these factors that they identified as responders or non-responders was high dietary fiber and probiotic use. So when they transferred these microbes into the mice, they either fed them a high or low fiber diet or gave them probiotics to mimic what they were seeing in their previous study. Then what importantly they did was they validated this in two other very large cohorts that had already undergone fecal microbiome transplant or immune checkpoint blockade. So what are the results of these data? Well, remember I told you to put a pin in that idea about diversity. They were not able to replicate that finding. But what they were able to replicate was that this response appears to be associated with the abundance of Rhamnococciaceae family members. They also determined that the probiotics might be contraindicated in patients with melanoma undergoing PD-1 therapy. They also indicate through the fecal microbiome transplants in the animal models that fiber may be beneficial in improving immune response. And I'm going to go into the details on that in just a little bit. But here's a nice kind of graphical overview of explaining what they're seeing in this study overall. So as fiber, dietary fiber ingestion goes up and probiotic use goes down, you see an increase in response to immunotherapy, whereas those in the lowest fiber intake and the highest probiotics seem to have the poorest response to immunotherapy. So what they were seeing, specifically in this study when they looked at the immune cells, they were seeing that the high dietary fiber was associated with CD8 T cell activation. And this seems to be a common theme that we see through a lot of these studies. And now I'm going to talk to you about what the actual mechanism is going on here. So Jennifer Wargo partnered with Romina Goldschmidt at the NCI to further develop and understand what the immune mechanism was actually doing at this time when we add dietary fiber to the microbiome in the context of melanoma. So what they did was they used the responder patients' microbes, or the non-responders. They inoculated germ-free mice, or they had the intact microbiota, and they fed them a high-fiber diet. And so the main question they were asking was, how does the diet affect the microbiome to activate the immune system in responders differently than in non-responders? And so the interesting thing that they found out here, they fed them actually dietary pectin. I had to actually call up Rosemina and ask her what it was, because I couldn't find what actually fiber they used, but it was dietary pectin in combination with immune checkpoint blockade. And what happened was that dietary fiber increased the abundance of a specific bacteria, Akkermansia, which we know is a mucin degrader and fiber fermenter. And what happened was these bacteria, not just Akkermansia, but several other bacteria, actually started producing cyclic dinucleotides, which activate the STING pathway. And so these products that were being made by the microbiome in response to seeing high dietary fiber were in fact helping to activate the STING pathway, turn on IF and gamma, and activate natural killer cells then to produce these chemokines to activate and recruit dendritic cells and recruit anti-tumor macrophages, which we didn't see happening in the patients that did not take the high dietary fiber and had poor response. So overall, what we're seeing is that high dietary fiber encourages the growth of microbes that produce molecules that activate the immune system to respond to immunotherapy overall. And we also see this happening with some of the chemotherapeutics as well. So to sum up what I've stated so far, we see the anti-tumorigenic effects of high dietary fiber, specifically fruit and vegetable dietary patterns. They increase certain bacteria that are capable of producing molecules that activate and recruit immune cells that help trigger and respond to immunotherapy, specifically CD8 positive T cells and natural killer cells. On the opposite side, where I think we still need some more data, is what is the effect of a Western dietary pattern? So high fats, low fiber. So we assume that what's going to be happening here is that these types of products being made and conferred by the microbiome are further producing more DNA damaging agents or reactive oxygen species, and maybe reducing the efficacy of immuno and chemotherapy. So now I'm going to talk about what do we actually need to move forward in the field to get to the point where we can do precision nutrition therapy and microbiome therapy. So first I want to lay out some of the evidence that we have, I think, from pretty good trials that have at least tried to do a good job of introducing different dietary patterns or different dietary interventions in cancer patients. So the first one I'm going to talk about is one that was published from an Italian group in Cancer Discovery, I think about 2 years ago. And what they did is they tried a fasting mimicking diet. So a fasting mimicking diet is just essentially a very low calorie diet where you have about 600 to 300 calories about 3 to 5 days before you start immunotherapy or chemotherapy, and then you start your therapy, and then you repeat the cycle before the next therapy session. Now, the critical component here is that they had multiple clinicians on call 24 hours, plus a nurse practitioner, plus a dietitian for the study because it was a phase 1 safety trial. And so it was an all-comer trial. They recruited over 100 patients with multiple different types of cancer. Again, it was just looking at safety at this point. So in a subarm, they were looking at early-stage breast cancer and melanoma. And of course, like they said, this study was not designed to be powered for efficacy, just safety. But what they went ahead and did is looked at some of the immune profiles. They did PBMC analysis. They also did RNA-seq expression from the tumors. And what they were noticing is that they were seeing signatures of improvement in cytotoxicity response, T cell recruitment, and again, we see this CCL5 also being upregulated as well. So indicating that the fasting mimicking diet could improve the immune system in response to treatment. This is another example of a well-designed trial. Their phase 1 trial was very impressive. It was only in a few patients in the beginning. They were looking at the effect of a metabolic ketogenic diet in glioblastoma. And it was very well done because they had a dietitian as well as several physicians to make sure that people were staying in ketosis. And they did achieve that through 85% of the study. And they had a really fantastic response rate. So they had a progression-free survival of 11 months and an overall survival of 29 months, which was unheard of for glioblastoma. So this is now advanced into a phase 2 trial, and we'll hopefully see what the results are. Hopefully they're equally as impressive. So the last one I'll leave you with, and hopefully I'll have data for this soon, is my trial where we're looking at the effect of diet and the microbiome on chemotherapy-induced diarrhea. And so a lot of patients suffer from this, especially those undergoing FOLFOX treatment for colorectal cancer. There's no clinical biomarkers, and some of the strategies that are currently used are not completely effective. So we do have some data showing that the microbiome could be involved, especially from this study where they're looking at radiotherapy in patients that were being treated, and they saw that there is a component of the microbiome, so you see a loss of diversity in people that actually had diarrhea from radiotherapy. So we've designed a study that is going to be looking at the effect of diet throughout the course of treatment and after treatment to see if there's factors related with the diet and the microbiome that are predictive of chemotherapy-induced diarrhea. So not only predictive, but hopefully we can identify mechanisms as well. And so that's ongoing, and hopefully we'll have some data for that in the next year or two. The thing that I'm most excited to tell you about, and coming back to my circle and my story that I told you in the beginning, I'm very passionate about helping cancer patients navigate their patient journey and helping identifying patients that can respond to treatment through changing their nutrition, but not only their nutrition, their mental and their physical health as well. So I worked with a company and I started Cancer Warrior Health. Cancer Warrior Health is dedicated to helping patients navigate their journey through cancer treatment by empowering them with solid, evidence-based nutrition, physical, and mental health guidance and recommendations. So the way this app works is we take a survey. They're all clinically and evidence-based surveys that clinicians and professionals use in their everyday practice. I worked with several different ones to develop these surveys. So we take a survey, an assessment, and then based on a decision tree, they get basic recommendations. This is not medical advice, very clearly stated, but it's recommendations that they can use to set goals. And then we track these goals, and then we give them feedback and do another assessment because we know their diet and their physical and mental health changes throughout treatment. And so we want to make sure that we're encouraging them to make better decisions and be proactive rather than reactive. So this is in the very early stages of development. We have a beta testing version right now, but my first step, as I stated here, is to be able to collect over 1,000 dietary recalls and histories from patients so we can start to get those data because we have very little data on how cancer patients eat currently. There was a recent NIH study produced in a workshop indicating this as such. And so we just lack hardly any data on how cancer patients eat because I'm sure you understand it's hard to get those data because they're so sick all the time. Okay, so where do I think we need to go moving forward? What are the questions we need to ask? So the first thing we really need to do is ask, how does pre-diagnosis dietary impact the structure and function of the microbiome at diagnosis? This will be really important to help to understand, can we and should we modulate the diet to impact treatment acutely? How does the microbiome modify dietary interventions, or chemotherapy, or immunotherapy interventions? What is the effect of diet on the microbiome structure during cancer treatment? And how does the microbiome at diagnosis affect nutrition status? So if you have a really poor, low diversity microbiome, maybe no matter what we do nutritionally, it's going to affect. So maybe we need to focus on the microbiome first and maybe amplify some other types of nutrition status later on. So the last thing I'll end with here is, where do I think we should go in the future? Where do I see us going? So hopefully in the future, we're going to optimize immuno and chemotherapy through either prebiotic or probiotics. They've already done phase 1 trials of FMT for immunotherapy, which seem to be beneficial, at least in this one study. Microbiome-based drug delivery, they've actually harnessed the effect of bacteria to produce immunotherapy through secretion of outer membrane vesicles. Something that my lab is working on, we're working on producing engineered probiotics that can target specific bacteria and eliminate them completely from the microbiome to improve cancer treatment response. And then last, again, my passion is to potentially have enough data to be able to perform precision nutrition therapy and microbiome therapy for cancer patients to hopefully eliminate or reduce cytotoxic effects and improve overall treatment outcomes for patients. And that's it. Thank you.
Checking. Oh, perfect. Okay. Before we take questions, I want to introduce our panel. So we have Krisztina Emodi. Krisztina, stand up. She is a nurse practitioner, Department of UCSF. She works exclusively with bladder cancer patients post-cystectomy and does a lot of probiotics education and patient education in terms of urinary tract infections and probiotic handling. And then we have our patient advocate, Gail. Gail, if you can stand up. It's great to have Gail here to give us a patient insight into probiotics and microbiome and things that we as physicians and researchers should be focusing on going forward. So if you don't mind, I'm going to start with the first question. And wonderful work, amazing work and presentation. So the studies that focusing on high fiber diet, a lot of them work in theory when patients or mice are in cages or patients are seeing providers who encourage fiber diet. Unfortunately, we know that as soon as patients are out of the clinical trial, they return to their low fiber diet fairly quickly. And high fiber diet is difficult for patients. Yeah. So what are some of the strategies based on your expertise that potentially can encourage patients to take high fiber diet independently?
Sure. So, I think it really starts with the clinician, because I think the patients really rely on the clinician and their advice in how to change their diet, and they see them more regularly, or the nurse practitioner, they see them more regularly. And I think it just takes the encouragement of the clinician and the team on a regular basis to encourage them to take on the changes in their diet. But the problem is we don't have enough dietitians usually at a lot of these centers to guide them throughout this journey and helping them improve their dietary habits. So I think it's going to be a combination of something like what I'm introducing here, like a health tracking app that they can be engaged with on a regular basis that actually gives them push notifications. Hey, this is something you can try to add to your diet, to add more fiber, to encourage more vegetable intake. So I think it's going to be a multifactorial approach. It's going to take the clinicians, the healthcare team to really be engaged in promotion. Maybe groups, when you have group therapy, promoting and discussing it then, so they can really start understanding how they can incorporate dietary changes into their diet, and the caregiver as well. So the caregiver has to be a complete component of this change in their diet, because a lot of the times they're not well enough to make the dietary changes on their own. Their caregiver has to be a strong component of that.
In terms of quantitatively seeing how patients are doing with fiber diet, I understand there's surveys and sort of recall in terms of what you ate. Is there any data in terms of blood or fecal sampling that you can do to document fiber breakdown or fiber diet?
Probably the thing that comes to the top of my mind, probably short chain fatty acids, but I don't think there's been enough strong clinical data. I would say during or at the beginning of treatment to be able to use that as a biomarker right now, but that's a great potential study to see if you could.
Any questions from this side?
I think it's really important from a clinical perspective, as you were saying. Speak up, okay. I think it's really important coming from a clinician or a clinical perspective because it's almost like an oxymoron. Our patients in the surgical setting went through usually neoadjuvant chemotherapy. They're really not feeling well. The caregiver involvement as they are helping them and preparing diet. But for us from a surgical end to actually prepare you for surgery, for cystectomy, having the low fiber, low residue diet for at least 30 days, people have an even more challenging time to eat. And after that, I have so many neobladder patients, especially along with some of the ileal conduits, they are oftentimes not able to eat certain things for nearly the rest of their lives. So creating this high fiber, incredibly resourceful microbiome within the diet, I think it's very, very challenging. My patients go home with a sheet from the hospital dietitian, which literally makes you eat fruit cups from, in sugar syrup. And I'm constantly struggling to sort of balance weight, nutrition, optimization of infections, UTIs. So it's all so much connected. And in our previous sessions, looking at survivorship to thrivership, it's we need all hands on deck.
Yeah, absolutely. I totally agree. We just have so little very good dietitian and nutrition support. I know MD Anderson has started using patient health navigators with a nutrition app that they help give their patients that seems to be helpful. So we just need a lot more different trials to figure out what's going to work for patients. Yeah.
I think Phil was first, not because I like it.
Do you think that do you think that high fiber is enough to turn your gut microbiome over to the point that you don't even need FMT? Like there's, I think we talked about a study a couple of years ago of like, it may have been like some sort of like native South American Indian tribe, where someone was measuring their microbiome and they saw that it completely fluctuated depending on what season it was because their diet changed.
Yeah, the hunter-gatherer tribes.
And you see these really rare bacteria during, and then they come out when they're eating fruits and nuts, and they go away when they're eating potatoes and start roots. So do you think that you even need FMT if you just have the right diet?
Well, that's a great question. I would like to say no, you wouldn't need to, but I wouldn't know unless you did the trial for it. It would be great to say you didn't need it, but I think it's probably going to be a both type of situation. We do FMT because we don't know how to change it better, right? But until we have better data with fiber, diet, and the microbiome together to figure out why it's working, all we have is FMT. I would like to get to the point where we could just use diet, but what you're saying when you were talking about dietary changes being next to impossible with patients, it might be that we have to do a more translational approach where we take stool samples, we dose them with the products that would encourage them to produce the metabolites that are making these improvements in immune response, and then we do the FMT with those microbes.
You're saying you modify the sample before you put it back in?
Yeah.
Oh, that's interesting.
So they have them in pills now, right? So you can do FMT pills. And that might be a better way to go than trying to force somebody to change their diet who can't, right? So
you imagine it wouldn't sustain though unless the diet changed.
Yeah, it would have to be like an ongoing basis type of treatment. Until they could change their diet, it would probably have to. I don't know if it would completely colonize. Like probiotics, they don't usually colonize. You just keep taking them.
And I think another potential avenue for research is the delivery of fiber. Another potential along the path of what you're talking is the delivery of fiber. A lot of our patients have to eat a lot of fiber-rich food, but potentially there's other ways of delivering fiber. whether it's shakes, IV infusions, or potential pills, that way we can just deliver it without having patients to eat tons and tons of food. I know they're not feeling well, in combination with FMT. And going back to FMT plus diet, one of the first slides that you showed in terms of obese mice and FMT, you really do require both, because one is not sustainable. If anything, if I had to hypothesize, it's probably diet that has more effect than FMT alone.
Great talk, and thanks for sharing your personal experience and how you got there. I'm Sam from Rutgers. My question was about timing of these interventions. I know you showed that 2-week, African versus American study, because, as you mentioned, we've done a lot of work improving recovery after surgery, and one of the things we do is a low-fiber ERAS diet. And I know that, these high-fiber diets cause a lot of bloating, which, even in post-operative recovery would be the same during chemo. So it seems like before any intervention starts is our best opportunity. Is there any evidence on, how early we need to intervene, or how close we can get to those treatments to have an impact?
Well, I would say, again, this is going to be very specific for each cancer type, as you're indicating, as you were indicating, because evidently fiber might not be the right thing for each type of cancer. Maybe it's a fasting mimicking diet for that type of cancer, because that would work best. I've seen immunonutrition work really well perioperatively for improving treatment outcomes or surgical outcomes. So I think it's going to have to be on a cancer by cancer, case by case basis to figure out what's going to work best for each patient and each cancer type. So while fiber might work really great for melanoma patients or kidney cancer patients, it might not be the right option for bladder cancer patients. It might be, like I said, fasting mimicking diet, or something that's less obtrusive, like you were saying, adding some type of immunonutrition to a shake to improve their surgical outcomes or treatment outcomes. But these are what I'm saying is that the possibility of changing the microbiome to improve treatment outcome is something we need to focus more research on, but it needs to be defined for each cancer type.
You sort of answered my question.
Okay.
It was, can this research be used in terms of prevention? Obviously everybody would love to prevent cancer. So your answer kind of disappointed me in that sense, because if it's specific to each type of cancer, then that makes it very difficult. However, we're finding out more and more. In fact, the woman sitting to the right of me is, looking at genetic triggers for cancer. So if genetically, if your genetic makeup has illuminated that you're likely to get bladder cancer, could this be used in a preventive mode as opposed to, once diagnosis occurs?
Well, I would say we have a lot more information on that, because there's been so much more well-designed studies, epidemiological studies, and some translational studies that show that certain dietary patterns, such as Mediterranean dietary pattern, seems to be, for most cancer types, preventative. Now, whether there is certain dietary patterns that are preventative of certain types of cancers, I think we need a lot more work to understand that. But usually the blanket statement I can give is based on the best data that we currently have, which is a plant-based Mediterranean diet seems to have the most effect on prevention of most types of cancers. But to answer your question, it could be that specific diets are required to prevent certain types of cancers.
So then, given that answer, how do we get the word out more effectively to the general population? I realize it's very, very difficult for people to change their diets, but on the other hand, almost each and every bladder cancer patient and survivor that I speak to, says, I wish I had known this, I wish I had known that, and in many cases is, I wish I had known that my diet could have, maybe even prevented this from happening, or could have lessened the impacts. How do we get the word out? I would imagine that you belong to a nutritionist society.
Mm-hmm.
How does that get done more effectively, maybe in combination with BCAN?
Yeah, That's a great question. I've been thinking about that for decades. And I would say if you look back, and this comes from my training at Johns Hopkins, we were talking about public health and how to get the message out about cessation of smoking. Well, it was a huge public service campaign, and it had to get a lot of campaign money behind it to thwart the tobacco companies. So essentially you're working against the big giant food companies that are promoting a lot of these processed foods. If you can have a campaign that's similar to the anti-smoking campaign, we might actually make a difference in changing people's dietary habits, but it has to be at that level.
Sure, and I'd just like to offer that I'm sure that BCAN is there to join whatever forces there are. So that's what Think Tank is all about, is trying to create synergies. Yeah, thank you.
Yeah, the more clinicians we can get talking to their patients about, preventative measures, including diet, it would be great.
Yeah, that was a fascinating talk, so thank you very much for that. When I was diagnosed with bladder cancer over 10 years ago, my daughter gave me a lot of literature that addressed diet and cancer-fighting properties of diet. And one of the articles I remember reading was about Japan, where despite very high levels of cigarette smoking, I believe the levels of bladder cancer were very, very low. And one of the hypotheses was that they just drink a lot of green tea, and it is kind of widespread throughout that society. Is that just anecdotal, or has there been any kind of rigorous studies that have looked at that and have looked at whether there is that kind of connection?
Well, so there's been a lot of studies on green tea, and there's a lot of efficacy of green tea. But I think what they're seeing in that Japanese study, was it the Okinawa study? Probably. They were showing that it wasn't just the green tea, it was their entire diet and their lifestyle. They were very active people up until their 90s. They had a very, I would say, kind of Mediterranean-like diet, lots of fish, lots of vegetables on a regular basis, and again, being active constantly. So I think it was the lifestyle that they were living. The fact that they were drinking lots of green tea was just one component of that lifestyle.
Thank you very much. Very interesting. I have a few questions. So first of all, of course, fibers come from vegetables, but they also come from fruits, and it comes with a lot of sugar, right? And the example you gave about keto diet, it's not necessarily only about fiber, it's about avoiding sugar, switching from sugar to ketones, right? So a comment about that. And also, if the fiber supplements are also alternative to substitute fiber. Okay, so kind of two questions already. And the third question, so I'm just going to ask them all. So I'd never thought about the diversity in microbes. Diversity is good in everything, including in microbes, obviously. But if you can comment about that low diversity is bad, high diversity is good from scientific point of view.
So it's context specific. So for example, when I studied the lung cancer microbiome, you don't want a diverse lung cancer microbiome, or you don't want a diverse lung microbiome. You want to keep your lung microbiome pretty non-diverse. So in that case, diversity is not good. But I would say in majority of cases, it is good. Vaginal health, a high diverse vaginal microbiota, not good. So in some of these low biomass settings that don't have very many diverse microbes, keeping the diversity low is important, and specifically in those places. But yes, overall, having a higher diverse microbiome, and I kind of use it like thinking about the free market system when it works well. You have competitors out there that are small businesses that are fighting for resources like the microbes in our gut. But if you have a monopoly like C. diff, you ruin that free market system, right? So you don't share those resources very well. And so having high diversity keeps that system working correctly because it acts like a buffer. You know, we have a buffer system, it buffers through certain ranges. The microbiome, being diverse, has multiple different functions, both shared and specific. And so keeping that diversity large allows you to buffer against insults on the system. But yes, the fiber pills could be an option, but I think it has to, again, be cancer specific. Yeah.
Hi, I'm Kriti Mittal from UMass. Just had a comment going back to the thought on green tea. So we are part of ECOG, and ECOG has a green tea study in localized prostate cancer subgroup, and we're having a hard time accruing to that study. While it's not open at our site, I think one of the concerns is how the study is considered interventional, pharmacological intervention versus not, and we're trying to change how this study is being promoted as really to be a medical intervention study. We're having challenges with that. And so going back to, we have to join forces, not just to change culture like we did, with the anti-smoking campaign. It's also about how these trials will be designed and run, because how centers get credit for running an intervention study which has a medicinal intervention versus not can also impact how science moves forward in that field.
Dietary intervention studies are extremely difficult to do because cancer patients are so sick and they have so many complications. So thinking differently about how to do dietary intervention studies is going to be really key, like doing milkshakes or smoothies or pills in the form that's something that's easy to consume, that's not going to be a burden on them.
I want to make a comment about we had a wonderful session yesterday about carcinogenesis of bladder cancer and sort of the mechanisms. And one of the data was coming from dogs getting bladder cancer. And they looked at not only the duration of exposure to multiple carcinogens, but also the levels that were detected in dogs that developed cancer versus not. And it was fascinating to see that the dogs who developed cancer versus the dogs who didn't develop cancer had no difference in the total serum or urine metabolites of these carcinogens. So then we started talking about potentially it's not necessarily the level of exposure, tobacco smoking, pesticides, herbicides, it's really how you process them. And every person is different. Your enzyme levels are different than a person sitting next to you. So potentially one person can smoke for 5 years and be highly susceptible to bladder cancer. And someone could be smoking for 50 years. We all know those people. They're smokers nonstop for 100 years and are still doing just fine. So it's really your level of what we call phase 2, sort of detoxification of these toxins. And your gut actually has tons of bacteria that reverse the process. So depending on what your microbe is, you can actually take those detoxified toxins and reverse it and put it back into circulation. So yes, the answer to that question is yes, your gut microbiome for sure has influence in how you process those toxins and what your risk is for any cancer at that point. Yes. We're going to do 2 more questions, then we're going to move on, but we'll do lots of questions in between.
Hi, Brian Billings with the New York City Bladder Cancer Support Group. My question is, when we receive a diagnosis, it's unbelievably traumatic, and there's very little we can do in terms of decision making in the process. And I think nutrition was one, and I certainly grabbed onto it to give myself hope. And my question to you is, does mental health play a role in your microbiome? And how could that, do you believe, be addressed differently with doctors in this process?
Thanks for that great question. Absolutely, mental health is a key component. There's been quite a few pretty well-designed studies now showing that your microbiome impacts your mental health. They've actually started doing FMTs and other types of probiotic interventions with people who have mental health issues and seeing dramatic improvements, which is really amazing because our microbes do produce chemicals like GABA and also induce GLP-1 expression in the gut that affects our brain. And so I think it's a big component of how your gut microbiome talks to your brain and your mood. So what I'm saying is that when I was talking about my app, I realized I couldn't just focus on nutrition. I had to focus on mental health and physical health as well. Because if you're in a very poor mental state, you don't want to eat healthy. You want to eat comfort food. I started doing that when I was initially diagnosed until I realized I needed to shape up and figure out how to change my diet. And once I started doing that, my energy levels dramatically improved, my mental health improved, and I think it was probably a combination of the changes in my diet plus it's probably the changes in my gut microbiome over time. So I think to answer your question about mental health, I think the combination of dietary changes and plus having people that were specifically trained on your healthcare team to intervene either personally or in a group setting is a key component of allowing you to change your diet or change your lifestyle to improve your cancer treatment response. Does that answer your question? Okay, perfect.
Hi, Lee. Thanks for a great talk. Vanessa Hale at Ohio State University. I'm intrigued by the fasting mimicking diet results and curious about your experience with adherence and tolerance to the diet.
Yeah, so I would say I wasn't part of that study, but I know that it took a lot of effort on their team to get those people to adhere to that diet. And I think they probably recruited people who wanted to adhere to that diet, right? So it takes a lot of effort with clinicians, dietitian, nurse practitioners to be involved but I think the thing I like most about the fasting mimicking diet, if it proves efficacious in more studies, is that while it's not like a lifestyle change, you're only doing the dietary fasting mimicking diet for 3 to 5 days around the time of your treatment. If the patient's BMI is over 20, which was the criteria, and they can tolerate it, then it could be a way to improve their treatment response. It seems to have the same kind of effects as we were seeing with the dietary fiber. But again, it's going to take a lot more phase 2 and larger trials to make sure that that's actually going to work for cancer patients. But it's at least it's short term, and I think maybe cancer patients could handle that for 3 to 5 days prior to treatment or surgery.
Thank you so much.
Thanks.
We're going to move to our next presentation, which is going to be me. So you get to hear me. So Dr. DeRosa unfortunately could not make it. She was presenting in microbiome as biomarkers, but we have great results in gut microbiome from Philip Bosch Lab and myself that I would love to share. So my name is Laura Bukavina. I am assistant professor of allergic oncology at Case Western, and I'll be presenting on our results in gut microbiome in bladder cancer patients, as well as our results including what happens during neoadjuvant chemotherapy with your microbiome. So we want to first discuss what the differences are, talk about potential ways that microbiome could affect your chemotherapy response before your surgery, and talk about what we think is the future direction for a lot of these trials. So we want to be, when we do microbiome trials, I do want to say that we want to make sure that we have hypothesis-driven work because microbiome can be very labor-intensive and very messy. So prior to going to a lot of microbiome studies, we all need to have a hypothesis of what we think is going on. And my goal today is for you to understand why we did it and where we're hoping this is going to go and benefit our patients. So there's different ways that your microbiome can really modulate your response to immunotherapy and chemotherapy. So if we talk about immunity, and we've had a great introduction to a lot of these topics, so I'll just sort of summarize it, is your gut microbiome has innate ability to really modulate your immune response through either T regulation or CD8 infiltration into tumors. There's also multiple byproducts, what we call metabolites, including short-chain fatty acids, that have been shown to be anti-inflammatory, anti-tumorigenic, as well as improve outcomes with immunotherapy and chemotherapy. In addition, there are certain microbes that also affect specifically different types of therapies and on different cancers, including CTLA-4 therapy, which is influenced by Bacteroides, and bifidobacteria has been shown to really affect your anti-PD-1 therapy. So as we mentioned before, it's not only overall microbiome, but it's microbiome per therapy per cancer. So we can't really broadly apply melanoma findings to bladder cancer. And I think one of the things that I think is incredibly interesting and is understudied at all capacity across all cancers is how your gut metabolizes your chemotherapy. So we know some patients do great with chemotherapy and respond really well. Some patients have terrible side effects but respond really well. And some patients don't respond and have side effects. And what really drives the difference if we're giving the same dose of chemotherapy to all patients? And that really has to do with how you metabolize your innate way of detoxifying a lot of these medications, but also your gut microbiome and how it detoxifies it. So in really focusing on how your gut microbiome metabolizes chemotherapy so you can have adequate chemotherapy levels in your blood without the side effects. One thing I wanted to mention is that if studies looking at this, and there's not that many studies, they've noted that majority of strains can metabolize up to 76 different drugs. And out of the 76 different drugs, majority of the bacteria actually reduce your serum level. So that means you're actually getting less of the drug than what you're actually prescribed for. For example, when we look at Bacteroides, which is a bacteria that you'll keep hearing about in my presentation, Bacteroides is one of those which is known to be a mega metabolizer. So it metabolizes over 15 different drugs in previous studies. And as you can see at the bottom, although these are non-chemotherapy drugs, when they looked at diltiazem, it almost reduced the concentration by about half given high Bacteroides counts in your colon. So I want to introduce our study and talk about the results. This was a multi-institutional study, recruited a total of 152 patients. In addition, we recruited 37 healthy patients, and then we had partners because we also wanted to measure confounding of your microbiome based on your environment. So not only did we measure the difference between the cancer and the non-cancer patients, we also looked at the differences across time through chemotherapy, as well as potential confounding by what you're eating by measuring partner microbiome, including their changes through chemotherapy as well. This is just more of the same. So this is just a schematic of what we did. So we collected multiple rectal swab samples right before their cystectomy. And then our other cohort, which describes the changes of microbiome, was actually 3 different samples before their cystectomy, after their diagnosis, during chemotherapy. So we first focus on characterizing differences, and as you can see, the yellow is the bladder cancer patients and the blue is the control, which are the healthy patients with no cancer diagnosis. And even though there's a lot of overlap, you can see that there's clear populations of patients that have different diversity in their microbiome. When we looked at the total prevalence, which is the simplest way to look at microbiome, this is just looking at characteristics overall. This is sort of like, you have this bacteria, I have this bacteria. This is the simplest way to look at it. What we found is that patients with bladder cancer overall had a higher Bacteroides and a higher E. coli count across multiple samples. So then our true interest was to look at what bacteria quantify your response to chemotherapy. And what we found is that when we look at responders versus non-responders, we found that 12 out of 14 what we call complete responder patients, meaning at the time of cystectomy they had no evidence of tumor. So that means even though they had muscle invasive cancer to start, when they underwent surgery there was no cancer left behind that could be found. 12 out of 14 of those had Bacteroides counts what we consider to be very low. Unlike the patients who did not respond, those patients were more likely to have higher Bacteroides counts. And then more interesting finding, in my opinion, is that if you actually look at the Bacteroides throughout chemotherapy, and granted we only had 7 patients, so it was a limited cohort. However, if you look at those patients and you look at their Bacteroides counts throughout therapy, you actually see that they continue to rise. So anywhere between 20 to 40 prevalence increase of Bacteroides versus the patients who had a complete response, actually they dropped their Bacteroides counts. So then we wanted to see if we can potentially reproduce some of that data in our mouse model. And the way we did it is we do what's called the BCAN model, which is the mice are given a carcinogen for about 12 weeks. They develop tumors at about 16 weeks. We ultrasound, we confirm that there's tumor. And then we usually sacrifice the mice at 24 weeks unless they have shown evidence of cachexia or tumor decline. So following BCAN treatment, we also collected stool samples before they started this BCAN carcinogen, and then multiple week samples before they're being sacrificed. This is just an example of what a CT of a mouse looks like. We inject some contrast to look into the bladder. And once we know that they have bladder tumors, we just keep an eye on their growth. In addition, we also did ultrasound imaging to confirm tumor. And on the upper, you can see that there's a nice tumor on the right side. So what we found is that while the mice were getting BCAN, there were spikes of Bacteroides. So at 6 weeks, we consider it to be an immature gut. So it's still developing. It's sort of a small mouse. But we know that once they were started on BCAN, there were spikes of Bacteroides. And once the BCAN was withdrawn, you can see a sharp decline and really no more spikes of Bacteroides later on. So it's sort of, although it's not the same data as human data, it's sort of showing very promising results that this potentially could be reproduced in the mouse model. So then we did something mathematical, and we tried to see if we can predict based on the signature, so not just one bacteria or 2 bacteria, but using hundreds of bacteria that we found in stool, and see if we can predict based on a signature, your bacterial signature, predict your chemotherapy response. And based on that data from 2 different institutions, we're able to achieve this AUC of 0.95. So that means 95% of the time the model was accurate at predicting the response or not. However, this is just based on those 150 patients that we do have to validate it in other studies and other institutions. It's promising, however, this needs to be validated. So if you translate that to what the sensitivities and the specificities are, which is sort of how we look at X-ray images or any test. If you take that data and you translated it, then you have a sensitivity of 70% and a specificity of 80%. Now, if you look at any other biomarker right now available, including your tumor mutational burden or specific mutations within tumors, nothing really has been shown to be predictive for chemotherapy response consistently well. And this is promising data. Again, it needs to be validated. I don't want people getting too excited. I know Phil is there, let's validate it. But this is fairly promising in that we can actually predict based on your signature from stool, we can predict who's going to respond. Because the first step is prediction of response, the second step is modulation. If we know you're going to respond, what can we do to make those people who are not going to respond and change their gut microbiome to match yours? So then we looked at potential mechanism. One of them was short-chain fatty acid. We had 20 patients in this analysis. And again, we looked at potential correlations between the gut microbiome signature and the short-chain fatty acid and looked at the clinical response data. So even though there was no statistical difference between the short-chain fatty acid in the stool and the plasma, we did stool and plasma levels, we did find that there was a correlation between lactobacillus and enterobacter with higher levels of propionic acid, which has been shown to be associated with clinical response to therapy in other studies. So I do think we need to continue this further. I think 20 patients might not be enough, and I do think there's some signatures that we need to continue to explore. So overall, I think there's very promising data showing that your signature in gut microbiome has the potential to predict your chemotherapy response. In addition, by knowing this signature, I think we can also modulate people's response. I'm very interested in additional aspects of the study, and that's looking at metabolism of chemotherapy. So taking people who respond and looking at their chemotherapy metabolites to see if potentially they metabolize things differently. Thank you for everyone in the lab. Thank you for Phil for being my mentor through this project, and I'll take any questions.
I want to ask a question really quickly. There's been a lot of studies, mainly in GI tract cancers, and I guess some other cancers as well, that this signature, mainly of Fusobacteria, keeps showing up over and over again. Usually it's in combination with Streptococcus and Campylobacter and Veillonella. And I was wondering if you saw any of these similar kind of oncomicrobes hanging out together in your bladder cancer, because one of the reason I ask this is because, while I said that we're developing an engineered probiotic to eliminate one of these oncomicrobes or pathogens, my doubt is that that's going to be sufficient, because what if they're, complicit microbes that are there along with it? You're just removing something and the other ones are going to come and take its place. So I just wondering what kind of signatures you saw.
So we saw so there were 15 different bacteria, and when you look at this signature, there is what they call coefficient importance. So if the higher the coefficient of importance, the higher the contribution of that specific bacteria to the model. Mm-hmm. And what we found is that there was really not one bug that specifically dictated the highest importance. They were all like 0.15 out of 1, which is sort of low. But so I suspect it's probably like a codependent mechanism on multiple bugs. One of them was Oscillobacter was, I think, number 1 in terms of the importance. And then the lowest was Ruminococcus. Yeah, so it was out of 10, it was on their list as well. I don't remember all 10 of them. I can pull it up. I can show you.
Laura, thanks for that great data. It's really exciting. I had a question. You had seen Bacteroides spikes over time in your mouse model, but it's continual BBN and same diet. Do you have any thoughts on what's driving those spikes?
I'm not sure what's driving the spikes, honestly. Anytime you collect anyone's samples from time to time, there's going to be sort of these small differences. So I think potentially that. We didn't change the diet. We adjusted for cage, so they weren't, there's not a lot of confounding in terms of that. The BBN was stopped, and after that, we've stopped even seeing those spikes. So I don't know if it's an adjustment. Potentially there's a high Bacteroides, and then potentially adjustment of other bacteria that lowers that count, and then potentially another spike, and then they lower. It would be interesting to go back and see if those spikes correlated to the change of BBN, right? Because the BBN, they drink for a week, and it's from the same bottle. So I wonder if if you collect stool later on, you're going to have that lower, versus if you just give them fresh BBN, it's going to be higher. So it might be something that we can look at. Yeah.
Thank you, very interesting. I wonder if, so I don't know about mice, I will ask about humans. Mm-hmm. So I assume that mechanism is related to activation or inactivation of chemotherapy drugs, right? In a gut microbiota. Yep. Okay. And just to clarify, your swabs were before chemotherapy, right?
So the ones who are responders and non-responders, it was multiple times.
Multiple times.
Yeah. Okay. So it was 3 before, during, and after chemotherapy.
And which of these points were most informative for your?
So between before and after, there was really not a big change. in Bacteroides counts, there was some fluctuations. The biggest difference was the responders to non-responders. So if you're going to be a complete responder and we swab you at the beginning, we swab you in the middle, and we swab you right before therapy, you're sort of that first Bacteroides count, and if it's low, that's most predictive for the response.
Okay.
If your first Bacteroides count is super high, we know that that correlates to poor response. But if you are poor response and you start high, your Bacteroides counts will continue to go up. Versus the clinical response, your Bacteroides will stay stable or they'll decrease. Again, that was only 7 patients in that arm. The second question you asked is the bigger cohort of 150 patients. Out of those were 40, I think there was 42 patients that received neoadjuvant chemotherapy. Those patients are all swabbed right before cystectomy, after chemotherapy. So we had 2 different cohorts to look at.
Which is interesting because it does show that this microbiome profile is very stable regardless of the changes of the diet, regardless of the treatment they received, which is, resilience, at least from the side of all microbes.
Right. Yeah. And their partners, I didn't have the partner data, but if you look at their partners, it could be an argument made that, you're in an environment, Bacteroides kind of spikes as the other bacteria, but if you look at their partners, their microbiome didn't change either. So we knew it wasn't because of some sort of dietary change at home, for example, that was potentially influencing it.
And if you have any data about urine microbiome, is it a thing? Is it not a thing? And how does it correlate with the gut microbiome?
So Bacteroides is present in urine. That's going to be some of the data that we present tomorrow at the Translational Summit. For the urine, actually, the lactobacillus is the one that's most predictive for a response. And I think that's probably just like a completely different mechanism. It's localized, versus this is probably a drug metabolism in your gut.
Okay, thank you.
You're welcome.
Any other questions?
2 questions that are somewhat related. So if you get IV chemotherapy, what is the biologic mechanism of the bacteria in your gut getting exposed to do that metabolism? And I guess relatedly, I can kind of conceptually understand the immune alteration as potentially talking about an alteration in response to IO, but specifically in response to neoadjuvant platinum therapy, can you comment a little about how it affects platinum in particular?
Yeah, we've had multiple thinking discussions with Phil about this already. So if you are getting IV chemo, it goes to liver for phase 2 biotransformation. It gets excreted into your bile acid, into your small bowel and your large colon. The bacteria in your colon and your small bowel then have the ability to remove that glucuronation group and put it back into circulation as an active chemo drug, which means you're actually getting the higher dose, or they can actually degrade it further into more toxic metabolites. In terms of the immunotherapy, so the bacteria in your gut actually stimulate your Treg, depending on CD8 or Treg function, and they actually translocate. There's multiple evidence showing that Bacteroides, E. coli, probably a couple other ones, actually translocate to surrounding lymph nodes, which increase your Treg function and CD8 function depending on which bacteria.
I was just going to sort of liken it to, if anybody's handy or a plumber, if you have a septic system or a finger, what's it called? Where it just drains into the yard, and all your septic and your toilet, and the soil bacteria, clean your water back out, and then it goes back. I think it's kind of akin to a septic tank in your gut.
Yeah, it's like a big oil spill. What do? Throw some bacteria on it. It's going to degrade it. Actually, Pseudomonas probably.
So I had a question about the, we were talking about it earlier, and I'm curious, since you see these Bacteroides spikes after BCAN therapy, have you looked to see, I think it'd be kind of a simple in vitro study to see does Bacteroides metabolize BCAN? Is that what's causing the spike?
Not that we know. We haven't looked at Bacteroides metabolizing BCAN, but certainly be very easy to check.
Yeah.
It does get metabolized, so when it gets excreted into urine, it turns into something called BCAN, which is, I think, something happens within the nitrogen.
Yeah. And there is a nice drug. It's called Microbiome drug database. It's very poor because there's not a lot of data on it, but that might be something that we can also look into.
Thanks, Laura, and congrats on this really interesting work. Okay, I got to ask a couple questions as a layperson relatively to microbiome. So we talked about, and you explained nicely how IV chemo and, the gut microbiome can basically impact the delivery of active drug.
Yes.
That's what we're saying, right? But you also mentioned urine microbiome and lactobacillus. So are these sort of predictive markers? These are baseline markers of response? Or is that impacting the microbiome in the urine that then either has some role in response to chemo or doesn't? Are those sequential, or do those exist sort of simultaneously and in parallel? I guess I'm asking you to theorize maybe a little bit.
So it's hard to say. It's hard to say because that same chemo, it also gets metabolized and gets into urine, right? I mean, that's where you want it to be. So potentially it could be metabolized, and it could be lactobacillus. We hypothesize based on what we found is that lactobacillus is more CD8 dependent mechanism in terms of tumor regression than actual mechanism of like chemotherapy. So we think lactobacillus is more immune based, and Bacteroides is likely more mechanism in terms of chemotherapy degradation. But no one really has data, and this is just all hypothesis at this point. We see those things completely separate, and they're not prognostic, they're just predictive for response at this point. We also know lactobacillus is high in normal individuals and high in complete responders. Now, could that just mean that those people have a better microbiome to begin with, like just healthier to begin? Maybe it's a whole body phenomenon. They're just healthier, better diet, just better urinary microbiome, less infections, and that's why they're responding? Potentially. We don't know. There's very little data.
Did you look at the tumor microbiome in these mice?
Yeah. Phil has that data.
Because I imagine the gut microbiome is affecting the urine, it's affecting the tumor. So what if it's not even that liver, bypassing something more directly with the tumor microbiome?
Phil, you want to comment?
Yeah, so we took the bladders and isolated DNA and just did 16S on the bladders. We saw, and we've also we've also taken whole genome shotgun data from TCGA, and you can and Laura's done a lot of this too, where you filter out all the human data and see what the trash was that got thrown away in TCGA. And I think we actually did see, to Lee's point, some fusobacterium. I'm sorry, your question was about whether it's the tumor microbiome that matters or the gut or the urine. I mean, they don't need to be mutually exclusive. It's probably some combination of all three.
Yeah, that's the thing. I probably think that they are linked, right, to those spikes that you're seeing in women.
You think that the gut
could be directly,
But that's the assumption that the bacteria. So when we do 16S within tumor tissue, we're assuming that those things are alive, and they're not alive. It could be, they could have infiltrated and died inside a tumor cell, and that's it. So we're just seeing things inside tumor. However, and they can't metabolize anything when they're inside tumor and they're dead, right? So you can potentially say there's all this bacteria inside tumor, but then you can't really know anything until you do more metabolic studies, even within tumor sample and in the gut.
Also, how has your experience been, validating the whole exome with the 16S? Are you finding similar? Is it consistent?
So
we haven't
done whole. Yeah, I think he's asking if we've done like a whole genome to validate our 16.
No, but we've done like single cell sequencing and whole genome sequencing to look at similarities in terms of urine and tissue, and they're very similar. There's a lot of contaminants that you have to know based on what you do, what they are, but we find very similar bacteria that are very common in urinary tract, like Pseudomonas, Lactobacillus, Klebsiella, E. coli. We find them in prostate too all the time. But it's really sort of the level and the changes that we are more interested in.
I was going to ask Lee if maybe you know, has anyone tried to take tumor tissue and just culture bacteria from it and find, can you comment?
Thanks. Yeah. So Susan Bullman is probably the world leading best person that actually does this on a regular basis. She's had a couple of papers come out recently. I think most recently she looked at the tumor heterogeneity of microbes using RNA scope and combination with spatial transcriptomics. And she was able to actually culture out from fresh frozen tissue specific microbes, mainly Fusobacterium, and I think a handful of other ones. But she was able to actually get those out of the tumor themselves even after they were frozen. And yeah, it's pretty impressive work.
Just because the genetic material's there doesn't mean it's
alive.
Yeah, you could stain with some RNA markers maybe that would get at it as well. But yeah, the gold standard would be to be able to culture it out.
And the things that we are finding at culturable, like a lot of things that we find in sequencing data is not considered non-culturable because of the difficulties with media. But the things, at least Bacteroides, is culturable, anaerobic, but culturable.
Thank you, Laura. I had one question looking at our post-cystectomy population and how would potentially this bacteria correlate to long-term issues that we all have, given the mesentery is connected. It is my bread and butter treating complicated infections all the time, long-term. And I have, the same example with people who smoke or don't smoke. I have horrible colonizers where I'm constantly treating infections and trying to really optimize getting, staying off of antibiotics with probiotics, with diet, with different supplements to acidify the urine, et cetera. And I'm just wondering, in this population, how would that, you think, potentially correlate along with the lactobacillus expression in urine? Because I'm trying to do as much prevention as possible.
I think it's probably different depending on your diversion type. So probably the conduits versus neobladders versus Indianas are going to have a different composition in their urine. But if you're looking at the baseline fecal microbiome and looking at their post-infection risk afterwards, we didn't analyze the data. We certainly have enough patients to look into it. It might be something that's like clinically relevant for us to go back and look at these patients and number of documented infections based on their sort of baseline microbiome. I don't know any data on that, but I can definitely look into it. Yeah.
So we had all this urine microbiome data, and one of the residents actually went back through the patients who donated urine and pulled up all their surgical complications, thinking that if they had infections pre-op, maybe it would correlate with infections post-op. We didn't really find a connection, but it's hard. We only had like 50 patients, and you're trying to correlate. You end up splitting into a lot of different buckets if they had GI complications, urinary complications, bleeding complications, failure to thrive, which is a very, vague. So we were thinking along the same lines, but you have to have a lot of patients really to do that kind of study.
I'm just wondering that I haven't thought of it that way, like a lot of my complicated. Stand up so they can see. Oh. I was just wondering, I haven't thought of it that way, that maybe a lot of the complicated infections that I'm treating along the way, let's say a year or two out, I should start doing fecal sampling and start seeing what's the common denominator, because at this point I think I have about 1,000 patients under my belt post-cystectomy, and I'm kind of functioning a combination of ID along with urology. So.
I mean, it's not out of this world to think it's a piece of bowel that they're using for their diversion. So it's not out of this world to think that potentially this has an effect.
And the thing is, when you give that patient antibiotics, you're just upsetting the rest of their microbiome. You don't have,
Yes.
We sort of don't know what we're really doing.
That's right.
We actually did it at USC. It was small study, not enough patients, it was 30 patients, but we didn't collect gut. It was just urine, but we saw the changes in the microbiome, the native bladder, native bowel, and the divergent. And it was hard to see any signals, but yeah, we could do that on a larger scale.
Yeah, I mean, I think with 150 patients, I think we probably, considering the high rate of UTIs post, and most of these are conduits, we can probably have enough patients to do like a secondary analysis on that.
So I was curious, is there a common denominator among the offenders of infection that could be targetable, that you most commonly see that you see in infections?
Yes, I would say E. coli, Klebsiella, Pseudomonas, those are generally our offenders. And again, I have a very particular subset of patients who are constantly getting infections no matter what we do. And I have the other group that they've had a diversion 5 years and they never had a single infection. And so when we start ruling out strictures or basically all the mechanical things in urology that could potentially lead to urinary accumulation and not draining properly, then you end up with the rest of your gut.
Yeah, and I wonder once you get colonized and once it's seeded into the kidney, you have all this parenchymal seeding and urothelial seeding. Like, it is hard to get things out. We know that even if you have no clinical signs of an infection, you take a biopsy, there's always bacteria within tissue. We know that, so potentially that's the reason.
Yeah, and it's also really, I think it's challenging to translate this to primary care because oftentimes my patients live very far, and even though they have access to Epic, MyChart, telemedicine, and whatnot, if you become symptomatic on a Friday afternoon and you can't get a hold of a clinician, they also become potentially extremely sick in a very quick amount of time, or very fast. So we go from colonization to I don't feel well, feeling fatigued this morning, and next thing you know, by Sunday they're septic. And so the preventive part and kind of getting tools to clinicians on the ground, I think it's really important for quality of life long term.
I was just asking about the common offenders because the platform we're developing now, it's kind of like a plug and play. If we know which bacteria you're looking to get rid of, we can develop an engineered probiotic to get rid of it. Yeah.
This is what Think Tank is about, making those connections and finding people to collaborate with. I'm gonna introduce our last speaker. I'm very excited to hear Dr. Patricia Hibbert here. She's coming to us from Boston University. She is a chair of global health. She's done remarkable work in global health, but she's also has incredible insight on probiotics and clinical trials and sort of FDA approval for a lot of these and the road to LGG.
Well, thank you very much. I'm honored to be here. And I'd just like to explain a little bit, a little bit about what I have learned about studying probiotics, and I think it's very similar to the issues that you are mentioning. So my background may sound like I haven't figured out what I'm going to do with my life because I work in global health. I'm an infectious disease physician, so I was thrilled to hear all these recent comments about antibiotics, etc. But what really irritates me is the fact that most of the patients that I take care of who are immunocompromised, that means transplant patients, oncology patients, etc., most of them end up on antibiotics to prevent infection. It messes up their microbiome, and worse, when they get infected, then they get resistant organisms. And therefore, what I wanted to do was try and see if there was a role for probiotics to control all of this. So that's what my chaotic background is all about. So apologies, but I did want to take a microsecond to talk about what probiotics are and what probiotics aren't, and the consequences of that situation. So first of all, probiotics at the moment have a definition. It's a definition from the Society of Prebiotics and Probiotics, is that the bugs are alive when they are ingested or whatever, and when they're taken in adequate amounts, they will provide a health benefit. That's the definition. Comes from 2001. And it actually hasn't changed since that time. And what you will also note is there is no definition from the Food and Drug Administration, and we'll get to that in a second. First of all, most of these live organisms are bacteria. Some aren't, some are fungi, etc. And they have real names. So the one we often hear about is Lactobacillus. It has a species name, and therefore it ends up with a strain name, and we know it commonly as LGG. Another common one is Bifidobacteria. There are about 8 main genuses, genii, I'm not sure, but it doesn't matter, because these are the real names. Now, if you go to a pharmacy store and pick up a probiotic, maybe you'll see a real name. Maybe you'll see a made-up name. And that is a problem. So, probiotics have been becoming more and more popular over the last 20 years or so. Why? Not 100% sure, but a lot of things are going on. People are interested in healthy diets, right? People are interested now in learning about the microbiome and having a healthy microbiome. People are interested if somebody says, you're in trouble, you've got an unhealthy microbiome, can we switch it? That's what we've been talking about today. Then the big one. Can we treat a non-healthy microbiome? And then other reasons that probiotics are being used very widely is a lot of people are selling them and they're being promoted. So there are many reasons we're hearing a lot about probiotics. So where do you get probiotics? You all know. You probably walked past a CVS this morning or yesterday or whatever. They got them. So they're in pharmacies, they're in food stores, but what they are in is a huge range of foods. And the one that drives me bonkers is probiotics in bread. If you toast it, it's toast. I don't know. In grocery stores and pharmacies, some of these bugs are sold in dietary supplements, and we'll get into that in a second. What are the places probiotics are not found? You don't go to the back of CVS to the pharmacy area to get any probiotics. They're over the counter. Why? Probiotics are approved as a live biotherapeutic by the FDA if they were ever approved. And as of today, there is not one probiotic that is approved by the FDA. So you'll never get these in the pharmacy. What's the definition of a live biotherapeutic? a probiotic, that live microorganism, that does any of these 4 things: prevent, treat, mitigate, or cure. You use those words, ha, you're toast. You have to use a probiotic as a live biotherapeutic per the FDA. So how does the FDA know that you are using something to prevent, treat, mitigate, or cure? They say to you, what is your intended use? And what they won't allow you to say is, well, I just want to have a look and see how it works, and then hopefully we'll be able to move ahead. No, what's the real plan? And the problem we've got here is that mostly we are interested in preventing, treating, mitigating, or curing something for our patients. So, foods are also in the purview of the FDA. The oversight of foods is from what's called CIFSAN, or the Center for Food Safety and Applied Nutrition. And what you're allowed to say about foods, remember, you can't prevent, treat, mitigate, or cure. I hope you're going to remember this for the rest of your life, because these 4 words are absolutely making life very, very difficult. You are allowed to make structure-function claims. What's a structure-function claim? I have absolutely no idea. But it's things like, and you've seen this on the TV, this probiotic maintains bowel regularity. What the heck is that? Nobody knows. But it's a structure-function claim. Live biotherapeutics, commonly known as biologics, are regulated by the FDA Center for Biologics and Evaluation Research. And what this means is you can't study any probiotic unless it is manufactured essentially according to drug or biologic standards, which means that there can be no variation in the active ingredient, how the material is put together. And these strict manufacturing and regulatory processes are only for products that you can only market these if in well-conducted clinical trials you have shown that the probiotic is safe and clinically able to prevent, treat, mitigate, or cure. So, why the heck is the FDA doing this? Because there's increased use of many probiotics, so it's hit their radar. A lot of advertising over the years, and you'll occasionally see FDA warning letters going out to probiotic manufacturers who are recommending their product to prevent diarrhea, treat diarrhea. That is a medical condition. You can't do that. And they will tell the manufacturer, cease and desist immediately. There are also a lot of concerns about product quality, because remember, these are dietary supplements, so they don't have to meet these quality standards. So the first thing is, does that product you bought yesterday at CVS actually contain the bacteria it says is in it at the right dose? Are there other bacteria there that they kind of didn't think it was necessary to tell you about? And is the organism alive? Because probiotics are supposed to be alive, right? And then the kicker, what other contaminants are in these products? So this raises a lot of question about what we are giving our patients and what any of us may be taking. And clearly what the FDA is very, very concerned about is that most of us, and I think all of us in this room, are interested in people who have a compromised immune system, who may be able to benefit from the use of probiotics. And what the FDA sees is risk, sepsis on Sunday. My patients usually get sepsis on Saturday night, which is even more irritating, right? So could probiotics be therapeutics? Clearly, we've had a lot of discussion about this, and I don't want to go over all the details about how probiotics work. The bottom line is there are many ways probiotics may work. Do we really know what any single probiotic is doing? No, but in general, the idea of altering the GI tract microbiome, we've been talking about this a lot, by lowering the pH or inhibiting the growth of those drug-resistant organisms. That's how they could do it. We talked, Lee, you talked a lot about bioactive metabolites as well, so that's another mechanism. And most probiotics are as dietary supplements, remember, none are approved as a drug, can have effects on the endocrine and nervous systems, and you heard about mental conditions. There can be some effects there. So could they be therapeutics? Absolutely. But then they would be biologic, essentially biologic drugs. So why do I care? Because I really wanted to produce high-quality evidence about the effect of probably the most commonly used and widely studied to date probiotic, Lactobacillus GG, and see if it would help some of the patients I take care of. So the first idea was for any of you who've taken care of patients on dialysis or with hemodialysis or peritoneal dialysis, you know, a lot of them end up with Staph aureus, meth-resistant Staph aureus in their nares. And we have ointments that supposedly reduce colonization with this bug. Doesn't work too well. It turns out that LGG and Staph aureus like to fight. And LGG wins. How about we pop some LGG in the nose? Seemed like a good idea. And so the first question was, how would I get funding to do a high-quality clinical trial? Real problem, can't go to a dietary supplement company because they really don't have the resources. But NIH does. Therefore, I applied for an NIH grant to do that. I also take care of a lot of patients with cystic fibrosis who sooner or later end up with very resistant bugs due to millions of doses of antibiotics that they have to go through. And I really wondered whether or not lactobacillus taken orally there could help. And then this was in 2004. I knew I was going to get older, and I was very worried about what was going to happen to my immune system as I got a bit older. And I knew that flu was bad, and flu followed by Staph aureus as a superinfection was something I did not want to look forward to. So I wanted to then look at the immunomodulating properties of LGG to boost the flu vaccine, which doesn't work very well in the elderly. So all 3 grants were funded. And I got a call from my program officer at NIH that basically turned everything upside down. She said, oh, could you just give the I'm not sure where the slides went, but anyway, could you just give the FDA a quick call and ask if you need an investigational new drug application to do your studies? Of course. So I did a quick call to the FDA that ended up going on for over an hour, during which time I got a dressing down about the fact that I truly didn't understand FDA regulations, and that was true. But it was clear that all of my grants hit preventing, treating, mitigating, or curing. And the answer was, okay, you have to get not 1 IND, you've got to get 2 INDs, and you're not going to be studying this ointment because this isn't a manufactured product. It would have to be compounded, and we aren't even going to talk about it. So if you want to do the cystic fibrosis study and you want to do the flu study, maybe. But you can't do it the way you wrote the grant. You've actually got to do a first-in-man study. I don't know why they call it first-in-man. I'm not very happy about that either. But let's call it first-in-person study. And the bottom line is we are going to figure out whether or not lactobacillus GG, that is taken by probably 10 million or more people every day throughout the world, whether or not it's safe. And I'm going to do a tiny little study of 50 patients to learn more than what the rest of the world knows. Now, the FDA said, you got to figure out who the hell is going to manufacture the LGG to our standards, our chemistry, manufacturing, and control standards, CMC. They're as bad words as prevent, treat, mitigate, or cure. Because basically, unless you've got those controls, you can't even use the product. Bottom line is, I was very lucky to find one of the manufacturers who does manufacture about 10 million doses of LGG around the world every day to meet FDA standards. The FDA also says you have to study healthy young people between the ages of 18 and 49. And you need to design a study that is going to show LGG is safe. So the first question is, who's healthy? That's actually a complicated question. We ended up testing all of these subjects, not only doing a history and physical, but we ended up testing all of these subjects for about 50 known infectious diseases that would have made them, quotes, not healthy in terms of the FDA, and they couldn't be on any prescription drugs, blah, blah, blah. And then, guess what? If you're studying LGG versus placebo, and somebody's taking LGG because they can just go and buy it at CVS, that's a problem. So we had to actually test a stool and culture for LGG as far as that was concerned. And we also asked people not to take yogurts. That meant we got people who were not terribly interested in eating yogurts. Who are they? I don't know. But those were the rules, and that's what we did. So we did this study, and you'll never guess what we found. Do you think LGG was safe? Yes! And we actually did look at the effect on the microbiome, and honestly didn't find anything. This was 16S. We have done some metagenomics, but 16S, it wasn't terribly exciting. The bottom line is, we then were allowed to do a couple of studies in the elderly population, not to do the studies we had originally proposed. And then, having done all these studies, I really felt it was terribly important to publish the data, because I didn't want anybody else to have to do these studies over again. I nearly killed myself to get 2 of the studies published. And then the well dried up, because what most of the journals who were receiving this said, what the hell are you doing these types of studies for? Everybody knows LGG is safe. We're not going to waste time publishing it. They still are not published. And so when we go back to NIH and say, can we now move to the next level? The answer is, of course you can't. You didn't do what you said you were going to do when we funded you. You're out of luck. The relevance to the very exciting work we've heard today is, are you going to be able to move forward? Is this going to be regulated as a biotherapeutic? I think there's quite a risk. But the bottom line is, it is possible to get an IND. For the intravesicular administration, I think this is going to be tricky, but I hope it can go ahead. But my personal opinion here is, something's got to give. I don't think this stance is right and fair. I've fought the FDA very hard on this, and I have not been able to get any further. So right now, all my probiotic research is completely on hold. And what I'm scared about is, where are we going with some of the dietary studies? I did ask the FDA about a study I did many, many years ago, which was looking at how long should exclusive breastfeeding occur, because in the majority of the world, kids are fed for 6 months. Is that the right thing to do? Is shorter fine? I did a study of that. I told the FDA about it. My God, you should have told us about that study. You were trying to prevent diarrhea in children. Breastfeeding required an IND. And with that, I really hope you guys can move ahead. I think you've got a very exciting agenda. And I hope you don't have a story like this in 5 or so years. Thank you for listening.
Thank you. Thank you so much. I'm so sorry. We'll go for yogurt later. I think everyone now understands why it's important to have all sides of the story. I think we see the amazing research, right? We see the potential that we can have in many of our cancer patients, but we also need to know what the roadblocks are in this. And having someone like you come and talk about the roadblocks roadblocks. I think we can potentially use strategies now in collaboration with BCAN to talk about how we can get a lot of this dietary LGG treatments out there and start working with FDA and sort of come up with a plan at this point.
So I think I'm going to ask the obvious question. Is the solution then to play by the FDA's rules, or do we have to change the way the FDA, like, do they need a new CIFSAN or CBER or whatever those, like, do they need a new agency within FDA to look at these, like, to look at prebiotics, to look at probiotics, to look at, because there's, I mean, I've been looking, there's this bugs as drugs call for applicant, like, is everybody that applies to that just going to go through the same BS that you went through? Like, we're just shooting ourselves in the foot unless we solve this problem proactively.
Totally agree with you. I mean, the real truth is CIFSAN does oversee prebiotics and probiotics. It's just the structure function.
Yeah, the rules that we have to play by are too constrictive. So the question is, how do you change? Does this need an act of Congress? Like, how do you change the way they regulate these things?
That high, because I truly don't think this can be fought easily through physician groups. I mean, the only thing I can think of that physician groups have won on is fecal transplants, when they went nuts when the FDA decided they were going to do the same damn thing for that.
Well, the other thing is, this may be like a public, I mean, people want this, right? Like, it may be leading from behind all this.
Yeah.
So what I was going to say is I agree. I think it has to be political. I think you have to find a champion in your local region, maybe in Chicago or in your district, that cares enough about this story, that had a family member or a patient or whatever. It usually pull on their heartstrings. And a lot of the times they have discretionary funds that they will line item in their budgets regionally or statewide, and you can write a white paper and get funding that way to do the research. And then you have your political champion that will potentially help gain more political will to change the regulations.
One of the reasons that I asked to be on the panel was that I believe that BCAN has a real role, as well as the patient advocates, in being able to, if not advocate for a real change, at least start the leading from behind. One of the things that's really important is for doctors, researchers to know that patients want to participate in these studies, that the ick factor of fecal collection is not going to be a stopper for everyone, and that even if there isn't a full study that is directed on some of these areas, being able to collect data and to be able to go back and look at data in other research studies is going to be important. One of the things that BCAN does so very well is that they bring to the fore the kind of quality of life issues. Well, colitis, diarrhea, these are constipation. These are quality of life issues. We don't talk about them. For most of us, we've spent a lifetime avoiding talking about urination or defecation, and now we need to be able to do it, and we need to be able to work with that vocabulary. We don't have a really good vocabulary. We don't have a common vocabulary. What I'm describing as diarrhea is not necessarily what anyone else in this room would describe. There are, for example, the Bristol diagram of what fecal matter looks like. BCAN and researchers can start to have those conversations. These are conversations in care and in care settings that deserve more than a 20-second Are you feeling okay? Are you normal? So I think that there are ways in which we can start to rise the level of awareness, and that BCAN can certainly do a lot to help that.
Go around clockwise.
A final question. So is yogurt good for you, or it has to be intravesical?
I'm so sorry?
If yogurt is good for you, or it has to be intravesically delivered to be useful?
I would like to challenge all of you and invite you to join me as the Director of Education and Advocacy at BCAN to go further with this. I think we as a national organization have an opportunity here because it's hampering research. There's brilliance here in this room, and they're making significant change on behalf of patients. And if your experience is any example that everybody else is dealing with, I think we as an organization will work to try to change that. So you'll see some emails from me in the future, Dr. Hibbard.
I am very sure that there are at least 20 other people. I can name half of that list off the top of my head. But if you actually look at what NIH is funding in this area, it's completely stopped.
I'd like to add to that as well. I'm really happy that Stephanie is here because it's just so important in terms of the advocacy part. One of the things that BCAN does with Stephanie in the lead is to go and visit congressional offices on Capitol Hill, and then follow up with local offices. And I think that feeds into exactly what you were saying. I did it for the first time this year. It's a wonderful experience. Certainly would invite any of you to do that. And just as an example, going into one of the offices, I found out that the congressional staffer, like you were saying, Lee, had a relative who had gone through the cancer experience, not bladder, but it was close enough. And that personal contact is incredibly important. The second thing is, as you were mentioning, Dr. Hibbard, is collaborating with other organizations. I'm a groundwater professional, and we go to the Capitol Hill for that reason. But collaborating with several other organizations, civil engineers, and so in this industry, there's the American Urological Association, all these other associations that when you collaborate, then you bring the power to the office in Congress, and that makes a huge difference, I've got to tell you. One question for you, Dr. Hibbard, is has the EU and the UK, have they solved this problem? Can we bring that to the table, say to Congress, to say, hey, look at the EU, are they doing things better than we are in terms of helping researchers?
So the EU has different regulations, and the FDA stance on both Canadian and EU regulations are, it doesn't matter. So there's been a disagreement with the way these products are regulated. There are some basic similarities, and I think these are similarities we would all agree with. Let's make sure we're manufacturing quality products, that aren't contaminated, because that's only fair to everybody who's buying them, and they're not cheap. So the bottom line is everybody's got that sorted out. But the process in the EU has been easier than the FDA regulations.
That's what I mean.
Yeah. To be honest, I don't feel qualified enough to comment on those. And then I don't know where the UK is after Brexit. They seem to have got sidetracked on some other issues. This is not a top priority.
My point is that it might be worth this endeavor, seeing what they're doing better than FDA is doing, and try to bring that to Congress, for instance.
Yeah, Congress, but FDA.
No, I know. Yeah, it's got to go to
I'm no expert in how the FDA works or anything, and know you guys are doing great work. So I'm just thinking of this as an example. You know, there's a lot of data and FDA definitions, for example, for BCG unresponsive disease, and there are these trials where all these new agents are getting FDA approval for patients with BCG unresponsive bladder cancer. But in reality and practice, in a lot of places like at my institution, outside of a clinical trial, the most commonly used treatment is gemcitabine and docetaxel, which I know these are two standard of care drugs, but the combination of those two together never had any trials or NIH approval. It just was done, and people did research and showed great results that it gained enough steam and momentum that it's now being tested in an NCI, funded cooperative group trial. So it might not be good for me to say that you should go around the FDA, but I think as long as you guys keep getting great data like this, there's just going to be more momentum behind it, and it'll be easier to break through.
Well, they've absolutely refused at this point to define prebiotics and probiotics. This, you can Google it. There's nothing. Absolutely nothing.
Doctor, thank you for your talk. I'm curious, as you've studied this intensively now, if I may ask, have you made any changes to your own lifestyle? I mean, do you eat a lot of yogurt? Are you a vegetarian? Do you eat sauerkraut? I'm just curious. I saw you wore a mask, which I applaud, but I'm just kind of.
I'm an infectious disease doctor.
Right.
I'm still taking care of COVID patients, so I'm just careful. You probably noticed I moved away when I took the mask off. When I was doing my studies, I felt that I should not eat yogurt at all. When it got to the point I couldn't even get the data published, I went back to eating yogurt. And that's probably about the healthiest thing I eat. I would dread for you to know what I really eat. I don't think you'd like it. But no, I'm not, looking at all the ways of trying to get it.
I can tell you I'm a vegetarian, and I drink lots of LGG. Trying to convince Phil to become a vegetarian too.
I just have one more comment. In clinical settings, I'm a big fan of the pre and the probiotic, as we talked about, helping patients. I'll speak louder. Okay. And over my own research coming through, I've been using. Oh, stand up. Okay, sorry. I came across the International Society of Pre and Probiotics, and I had no idea what I was doing. And looking at different strains and are my patients coming down with infections and whatnot, I came up with one probiotic that I've been using. And as you said, there's no regulation, there's no oversight for a lot of these. And I tried to find one certain something that would actually potentially work. And I'm not representing any companies or anybody or being funded. I've been using Visbiome, which has worked for me and for the people I work with. And it comes refrigerated, controlled with temperature controls in an ice box. And so I thought that was one of the best things I came across, and I'm just wondering if you have any thoughts on that or any other recommendations for the bladder cancer community, because when you go onto the International Society, the website, they actually list every single component and what conditions have been studied in clinical trials and data. So I was wondering if you had any thoughts on that.
So putting on my infectious disease hat, there are definitely some strains that are higher quality than others and less likely to be contaminated. But given that many of the people we treat are immunocompromised, I just want to be sure that the strain that is in a particular probiotic is one that is treatable. I don't want it to be a pan-resistant organism. And there are actually some, quotes, probiotics that are pretty pan-resistant. Enterococcus is the group I'm very, very worried about. So honestly, I'm not 100% sure what's in this particular product, but that is something that I care about, because if we expose an immunocompromised patient who then actually gets a systemic infection, we've got to be able to treat it. So that's one thing I personally feel strongly about. I think in general, I'm always hearing stories of people saying, this probiotic really worked for me. And maybe it is working for many people. And in general, I don't think we've got a huge safety signal here. People are not dropping dead all over the world when they take probiotics. So I think in general, it's fine. I really just have to know that the infection is likely treatable. That's my only caveat.
Okay, I think we had an amazing breakout session. We're going to close it off. I think it's close to 12. We're going to finish off, and then everyone can grab lunch before the next session. But I want to thank our speakers. Amazing job, but this has been an amazing